Thank you for joining me for another edition of Parker Biology. Today we're going to look at fermentation and regulation. Most cellular respiration, when we discuss aerobic respiration, we are looking at a process that requires oxygen in order to produce ATP. When we look at the setup of the electron transport chain, we can see that oxygen is vital, waiting at the end of the ETC. It's highly electronegative, and it's helping to pull the electrons down the chain, ultimately producing water. Now with that, if oxygen is missing, the electron transport chain will not function efficiently. So the cell needs a backup plan, or anaerobic respiration. Anaerobic respiration includes glycolysis. It can happen whether or not oxygen is present. And fermentation is going to couple glycolysis with additional reactions to help regenerate NAD+, or that electron acceptor. The two common types of fermentation that we're going to examine are alcohol fermentation and lactic acid fermentation. Each of these is named for the product that results. Here we can see the two examples of the main types of fermentation. And the overall gist is that glucose will be broken into two pyruvates. No matter what, glycolysis will occur. And this results in two ATPs generated via substrate level phosphorylation. Well, if oxygen is lacking and the ETC cannot function optimally, glycolysis will still occur. But if it stopped there, if it just ended with those two pyruvates, sure, the cell would get some ATP. But notice that the electron carriers would all be filled up. NADHs would have all gained all of the electrons that they could. They would all be reduced. And if all of them were reduced, there'd be nobody left to accept new electrons. And glucose wouldn't be able to convert into pyruvate anymore. Without the electron acceptor, electrons couldn't be ripped off, and this conversion couldn't go on. The cell in that case wouldn't be able to make ATP anymore. So instead of just stopping there and allowing the cell to die, glycolysis gets coupled with another reaction where the pyruvate's going to be further converted, and ultimately NAD+ will be available again. This allows for the recycling of the electron acceptor. In alcohol fermentation, the product is ethanol, whereas in lactic acid fermentation, the product is lactic acid, or lactate. Over here we see glucose converted to the pyruvates, and again, an additional reaction allowing for more electron carriers. Only a couple of ATPs made this way, but it's definitely better than the alternative of the cell dying. The type of fermentation that occurs depends on the type of cell that you're in. If we're looking at a yeast cell or other microorganism, they're more likely to carry out alcohol fermentation, whereas lactic acid fermentation occurs in our muscle cells. This is why when we work out too hard and our cells are deprived of necessary oxygen, they kick over into this anaerobic lactic acid fermentation option. Lactic acid can build up, and this may be a cause of some of the burning sensation we get during a really vigorous workout. This is a great diagram reminding us that no matter what, glucose will be converted into two pyruvates via glycolysis. It does not require oxygen, and this is just happening out in the cytosol. I love that we've got this fork in the road, that if oxygen is available, the pyruvates will enter into the mitochondrial matrix. We'll see the oxidation into acetyl-CoA, continuation into the Krebs cycle, or citric acid cycle, and finally then the electron transport chain, which is a lot more efficient. We'll see a much higher number of ATPs generated here. However, if there is not oxygen present, fermentation will ensue, leading to the production of ethyl alcohol or lactic acid. Here's another great diagram that reminds us about the nature of organic molecules. Yes, we typically draw out cellular respiration with reference to starting with glucose and getting our ATP from that particular organic molecule. However, we also know that energy does come from other sources. So what this diagram does is it shows us that yes, we can eat proteins, we can take in amino acids, and that those can enter into some different parts of these cycles. We also know we can eat fats, break them apart into glycerol and fatty acids, and those also can play into these metabolic pathways, ultimately resulting in some ATP production. We don't always talk about starting with those, but we know we can eat foods other than just carbs and get energy. The final idea for this unit is regulation. It's important that our cells only carry out processes when it's actually necessary, which means if we need ATP, we should make more ATP. If we have a surplus of it, though, we can probably relax for a while and our cells can slow down a little bit on this ATP production. It's important to keep in mind that each step along the way of cellular respiration is catalyzed by a particular enzyme. And our cells use this fact and they regulate the enzymes at strategic points along this catabolic or breakdown type of pathway. One of these strategic points in the metabolic pathway happens to fall within the process of glycolysis. Here we see again glucose being broken into those two pyruvates, and there are actually 10 smaller steps within glycolysis, each catalyzed by its own enzyme. Here we see an enzyme that's highly subject to regulation. The name of this enzyme is phosphofructokinase. Now, when the cell has plenty of ATP, when there is a surplus of energy, we do not need glycolysis to proceed as rapidly. So ATP can act as an allosteric inhibitor. This is feedback inhibition. This product comes back around to the enzyme and says, hey, enzyme, you can relax a little bit. There's plenty of me. However, as the cell starts to deplete this energy source, the enzyme will have to turn back on. Another way we see regulation is as a result of the citric acid cycle. There's a product called citrate that comes out of here, and if the cell is making a lot of citrate, it means it's doing a lot of this cycle, and ultimately a lot of ATP is probably being produced. So as citrate starts to build up, it can also come back around to phosphofructokinase and tell it can slow down. All of this is going just fine. We mentioned a second ago that if ATP starts to be used up, if there's not as much of it available, not only will it leave phosphofructokinase alone, it'll stop inhibiting it and allow it to do its job. But remember that ATP will be reduced down to ADP, adenosine diphosphate, and if the cell gets really desperate, ADP breaks even further down to AMP, or adenosine monophosphate. And this is a really clear sign to the cell that says we're pretty desperate, we're down to the bare minimum, we need some more energy. What's great about this is this adenosine monophosphate actually stimulates the activity of phosphofructokinase. It will help turn up the activity of this enzyme and say, hey cell, we're desperate, make some more of this stuff, keep this pathway going to eventually get some more ATP. Hopefully this little recap helped some with fermentation, the idea that yes, we can take in a variety of organic molecules and still get ATP, along with regulation. Thanks for joining me for Parker Biology.